So we are going to start with Chapter 1, Introduction to Genetics. This chapter has a few things that you're going to have to study and know, but mainly it's an introduction to the course and to the textbook. So I will point out a few things that you'll have to memorize and really know, but it's not a very long chapter, a very long lecture. Now all of the chapters begin with what I call a vignette, which is a little story, typically has to do with a certain gene or a certain allele of a gene, mutation that causes some phenotype or some interesting thing. And the chapter 1 vignette is called Albinism Among the Hopis. And the vignette will answer the question, how does the Hopi culture contribute to the high incidence of albinism among the Hopi tribe? So there's a story about it, and you have to know the story, what the gene is and what the story is for each of these. There are a few that aren't specifically about certain genes, but you need to be able to say in 2 or 3 sentences the gist, the main important stuff that's in the vignette. So as you're reading them, I would try to summarize it as concisely as you can so that when you go back to study, you don't have to read them all over again. But the vignettes are going to be your responsibility. So chapter 1 is partly about making a case that understanding genetics is important. And I think it's hard to argue that, that genetics is not important in today's society. The technology is moving along at such a pace. If you don't know any genetics, you're going to be a little bit at a disadvantage in the way things are going. And so you probably know a little chromosome theory, which states that genes are located on chromosomes, and chromosomes are passed from parent cell to daughter cell during cell division, and that those genes encode proteins. You'll remember transcription and translation is what gene expression is. So when genes are expressed, there are certain proteins that are made from those genes, and they create different phenotypes. They're responsible for all kinds of phenotypes. In this slide, you have bone structure of the hand, for example. So you have a series of genes, a lot of genes that are contributing to correct bone structure of the hand, and then certain mutations that could lead to a deformity shown in the bottom of part A in this picture. Then in part B, you have chromosome 5. It shows the location of some genes that are responsible for certain phenotypes. And remember, there's always a normal copy of that gene and an abnormal copy. So a normal allele and a mutant allele. So when we talk about an allele for dwarfism, what we're saying is if you have the normal version of that allele, you have a normal height. If you have a certain mutation of that allele, then you have dwarfism. So the alleles are typically named after the mutation phenotype. So a dwarfism gene is something, they're really talking about an allele where there's a normal allele and a dwarfism allele of the same gene. And so anyway, these have been mapped to different chromosomes. They're showing chromosome 5 here. And when there's a bracket, it means they don't know exactly where the gene maybe is, but they've narrowed it down to a certain section. We will talk later on about what the stripes mean on the chromosome. You may recall that the center point of the chromosome, the primary constriction, is called the centromere. So that's right here. That's the centromere, shown in gray. But anyway, we'll be talking more about that as we go along in the course. We will talk a little bit about embryogenesis and development, although that becomes a pretty specialized area of genetics if you study the genetics of development and embryogenesis. But one thing that's interesting about it is that as we studied more and more organisms, what we realized is that it's not it's not only what genes are in an organism, because a lot of organisms have very similar genes. Even fruit flies and humans have a surprisingly high proportion of genes that are pretty much the same, but it's not what the genes are necessarily, but how they're regulated. So in the picture you have a Drosophila embryo that's been stained for the expression of Hox genes. Hox genes set up the segmentation structure of the embryo. And so you see these stripes. There's 2 different genes being expressed in different locations. So the blue stains one type of Hox gene that's being expressed in these alternating stripes. and then the red is a different Hox gene. And and then the white means no expression of those genes. Now, every cell in the organism has a copy of every gene, but not every cell is expressing those genes all the time. So gene regulation means the decision that a cell makes regarding what genes are going to be expressed in that type of tissue or that type of cell, when those genes will be expressed and when they will not be expressed, and at what level they'll be expressed. Because a gene can be expressed, at a low level to produce a small amount of the protein product, or it can be heavily expressed to make a higher level of protein product. So gene regulation is really involves a lot of aspects of the cell making decisions about what gene to express, how much, and when. Now, good or bad, genetically engineered, genetically modified crops are a huge part of our food supply. We don't know, to be honest, what the long-term effects of eating GMOs is going to be, but we're the experiment. So we'll find out, I suppose. And certainly it's led to people going, to kind of an organic movement, and you could certainly argue that that's at least safer or less risk, I guess you could say. We don't really know what the risk is for GMO crops. But it has allowed us to produce plants that wouldn't be able to grow in drier environments to increase the food supply. And so there are good sides to it. We just don't know what the health effects are. And then in pharmaceuticals, you have, for 40 or 50 years, we've been producing recombinant human insulin, human clotting factors for hemophiliacs, growth hormones, and a lot of vaccines and such are produced by recombinant DNA technology. So there's good and bad. There's definitely advantages and risks to genetic technology, and being able to understand the genetics is certainly an important part of being part of our society. Now, there's basically three, you do need to know these, the three areas of genetics, three fields that your book lays out for you is transmission genetics, which is Mendelian genetics, molecular genetics, which focuses on the biochemistry of the DNA and the RNA and the protein, for example, and population genetics, which overlaps with evolutionary genetics and the genetics of groups. And so we're going to start the course with fundamentally transmission genetics, starting in chapter 3. And then we start with molecular genetics around chapter 8. And then we only do one chapter with population genetics in one lab. So it'll mostly be a pretty even split between the fundamentals of transmission genetics and molecular genetics. There are 6 commonly used model organisms in genetic research, and you do need to know these. You do need to memorize the names, common names, and scientific names. And I'm going to pronounce them for you because the use of the language in science is one way that people judge you. And I would want you to be using the correct pronunciations. If you, for some reason, had to pronounce these in front of somebody who knows the correct pronunciation, you want to show them that you're at a certain level of your education with regard to these. So I'm going to pronounce these for you. And just listen closely and see if you can understand what I'm saying. So the first one is, the fruit fly is Drosophila melanogaster. That's not too hard. The bee is Escherichia coli, or E. coli, but Escherichia, if somebody pronounces the whole thing. That's what they're saying, Escherichia coli, but a lot of times we just say E. coli, that's a bacterium. C is pronounced Caenorhabditis elegans. The C is soft, so you say a Caenorhabditis, not Caenorhabditis. Caenorhabditis elegans, we often say C. elegans for short. Arabidopsis thaliana, that's the thale cress or mustard plant. Mus musculus, that's an easy one. That's the mouse. And then Saccharomyces cerevisiae, the baker's yeast. And these are the 6 top model organisms that your textbook author picks. I would say that's a good pick. Nobody would really argue with these 6 being the top 6. You could add a few more. There could be maize, corn would be maybe number 7, or maybe a zebrafish, but these are a good solid 6 model organisms that scientists wouldn't argue with, that have been used heavily and are the 6 that we're going to highlight throughout the course. So why do we study model organisms? I mean, in some level we study them just to understand how genetics works in different organisms, just for general knowledge. But ultimately, when we study them, we're looking for mechanisms that are in common with human. We're either trying to develop drugs, ultimately, or trying to develop crops, or trying to look for cures for disease. So in applied science, we're looking for things that we can connect to something similar in humans. So for example, zebrafish, which didn't make our top 6 model organisms, but would certainly be in the top 10. Zebrafish have been studied, and one example, just kind of a simplistic example, is that the melanocytes in zebrafish express a pigment that's it's not really black, it's dark blue. And there's a mutant for that gene that produces an orange coloration. And it turns out that that gene is one of a couple of genes that produces pigmentation in humans. So pigments are a combination of usually several pigments to give you different shades and different levels of orange, brown, black are typically your mixtures that you have. And so you have normal zebrafish have normal alleles for the gene for that pigment, where the golden mutant is the same. The gene has a mutation in it and it gives you this golden color. So it translates to a mutation that you would see in somebody with red hair, red pigmentation in the skin, or really more orange, but we say red. And then the normal pigmentation would be like the normal zebrafish would relate to the normal coloration of the other individuals in the picture. So now what we're saying is, though, that things that you study in one organism can translate to the same type of gene in another organisms. So at least 10,000 years ago, maybe more, we have evidence that farmers were using the concepts of transmission genetics. And what I mean is that they were doing selective breeding, animal domestication, artificial pollination of crops. And because these require manipulation of sexual reproduction, they are technically practicing, or they were practicing, transmission genetics, or the principles of Mendelian genetics. You might call it common sense. If you want to have a good herd of cattle, then you're going to pick your strongest bull and your strongest cow, female, and you're going to put them together in a pen and try to make sure that they mate together so that you get the strongest calves that you can get. So, but this is manipulation, and it's been happening for thousands of years. And so, in that sense, humans have been doing transmission genetics for at least 10,000 years, maybe more. But we really start genetics as a field of science, can probably only claim to go back a couple hundred years. So even though there are elements of Mendelian genetics in agriculture that go back 10,000 years, typically we go back to, the 1800s, something like that. Before the 1800s, there were concepts of heredity going back even to Aristotle, but nothing was ever, proven. There was really no evidence or experiments. There were different concepts, a couple that are presented here, and you'll read about them in your text. One is pangenesis, this idea that things they called humors, which is kind of like liquids maybe, were drawn at the moment of delivery of sperm or release of egg, that these things would be drawn from all parts of the body, and they carried the traits from all parts of the body, and then they would go into the sperm, and then the sperm would be released, or they would go into the egg and the egg would be released. And that led to the formation of the zygote, the fertilized egg. And so pangenesis involves this sort of concept of liquids kind of coming together into the gametes and carrying the traits from all parts of the body into the gametes. And of course that's wrong. And what sort of overlaps with the pangenesis concept a little bit is the concept of blending inheritance, the concept of inheritance of acquired characteristics, which you may have heard of in a Lamarckian discussion from an earlier biology class. But these concepts, of course, are wrong. But they were floating around, you know, for hundreds and hundreds of years, even before the Dark Ages. And so they were pretty well entrenched by the time Mendel came around, even though there really wasn't any evidence, I would argue, for it. And what eventually was shown is that the theory that is correct is called germplasm theory, but it wasn't possible to understand germplasm theory until you could understand, really almost until you could see chromosomes under the microscope, so almost 1900 before you could really see that. But the germplasm theory says that there's a special set of tissue that carries the same genetic information as the rest of the body, although only half the amount of genetic material in each sperm or in each egg, but those are the cells that form the zygote, that nothing is drawn from the rest of the body, but those are sort of the representative cells for sexual reproduction. And that's correct. So you remember from meiosis that sperm and egg each carry a set of human chromosomes, and that when they fuse together, that forms the zygote, which is the first cell of the new individual. So that's germplasm theory. But you had all these other ideas, the pangenesis, the blending, the inheritance of acquired characteristics, and also you have this other wrong ideas, the concept of preformationism, which this is a very famous drawing of a tiny little man. The homunculus is a tiny, perfectly formed man, or woman, I suppose, but it's always shown as a man, that's inside of a sperm. So in this drawing, it kind of indicates the importance they thought that the sperm had. The sperm carries, this was an idea, like I said, prior to, but prior to the 1900 mark, where they thought that maybe the sperm carried a teeny tiny little man, and then when the sperm is delivered into the uterus provided nutrients to grow that little man bigger and bigger and bigger, and that's where babies come from. So I guess there could be a girl in the sperm, but anyway, of course that's wrong. We know that's wrong. So you have these, what seem now to be strange ideas, but those were pretty well, people believed those. So you put the real beginning of genetics usually starts with Mendel, and he's in the mid-1800s. So up until then, you've got a lot of strange ideas floating around. But by the time Mendel came around, you already had a concept of cells and cell theory, because that came around in 1839. So you've got that, but you really don't have chromosomal theory. You don't know, at that point, he didn't know anything about chromosomes. He didn't know about genes. He didn't know about meiosis. So you're still, I would say, kind of working in the dark. If you were Mendel, you still didn't have much to go on. And what's interesting about Mendel, and we'll learn about Mendel in chapter 3, is that he was working at the same time as Darwin. Virtually the exact same time. And they published at close to the same time. And one thing about Darwin's work is that it had a terrible explanation for inheritance. If Darwin had had a better understanding of inheritance, his work could have gotten more acceptance, I think. But he didn't have any real good understanding of inheritance. And Mendel wouldn't publish until 1865. And even when he did publish, nobody really understood what he was trying to say, because it was so different from those wrong ideas that they believed in. But I put a crown on Mendel because he's typically called the father, not the king, but the father of genetics. So a lot of, for the most part, we really mark 1865 as kind of the beginning of modern genetics as a field. Now, like I said, in 1900, when you start, in 1902 actually, when you can start to see chromosomes under the microscope, and in 1900, when 3 scientists separately repeated Mendel's, not the same exact experiments, but very similar concept experiments, you have typically what's called the rediscovery of Mendel's work. So in 1900, De Vries, Correns, and Schermak individually, they weren't working together, individually did similar kinds of experiments. You may remember the monohybrid cross, the dihybrid cross, those types of experiments. And each of them thought that they had come up with something brilliant, which they had, but unfortunately they found, I guess unfortunately for them, they found Mendel's publication, and that means in science you have to give credit to the first person who discovers something. But it's called the rediscovery of Mendel's work because it seems like in 1900 the rest of the world was ready to understand what Mendel was talking about. And very quickly after that you have Sutton and Boveri, which did not work together, but they individually described the chromosome behavior of cell division, and this laid the basis for what is commonly called the chromosomal theory of inheritance. So look that up. Chromosomal theory of inheritance is important. It says that that genes are located on chromosomes, and chromosomes are passed from parent cell to child, or to daughter cell. So they're the basis of inheritance of the traits that are encoded by those genes. Now in 2001, maybe 2003, you have the completion of what's called the Human Genome Project, and we'll talk about the Human Genome Project in chapter 15. But that was really just the beginning of the modern age of genetics. So this picture shows how we used to do DNA sequencing, which looks really highly technical, but now it's even more advanced. But when you have the discovery or the sequencing of all the human chromosomes, you really, I mean, that was just the beginning. And in 20 years, what we've accomplished from that time till 2017 or almost 2018 is going to amaze you, I think. So the speed of technology in the field of genetics is just, like any technology, is just increasing. All right, so what are the things you need to know? What you need to know from chapter 1 is the wrong concepts of inheritance. So the preformationism, the pangenesis, the blending, the inheritance of acquired characteristics. You need to know that germplasm theory is part of the correct concept of inheritance. You need to know the names of the 6 model organisms, scientific names and common names. You need to know the 3 fields of genetics: transmission, population, and molecular. But then you also, as sort of what you should know coming into the course, is listed here. So these are things that you already need to know. Number 1, basic structure and function of prokaryotic and eukaryotic cells. What are the rules that govern the cells, the energy flow, the materials flow through the cells, gene expression, which is really part of that, but I've listed it separately here. Gene expression is just transcription, translation, just the general concept of how genes are expressed. Cell theory, which states that all organisms, all life is made of 1 or more cells. The cell is the smallest unit of life, and that all cells come from other cells by cell division. You need to know the relationship and the definition of genotype and phenotype. You need to know your basic cell division, especially your mitosis. I will not spend very much time going over mitosis. I'll spend a little more time on meiosis. You'll have a lab on that. And then the basic rules of evolution. So if you're not, fundamentally, I mean, in a general sense, understanding these things, then you're going to be lost in the class. So make sure you have these basics. If you need to brush up, that's certainly fine. There's tons of resources online, Khan Academy, all these things that you can look up yourself. But just make sure you feel pretty comfortable with these things before you get going too much into this course. Because I want to make sure that you're successful, and I want to make sure that you have the right foundation.